Hello friends, Ace Botany, next episode, Sexual Reproduction in Flowering Plants, fifth one. One more episode needed for this chapter, Sexual Reproduction in Flowering Plants, that is the last part. So here we'll be mainly dealing with, in this episode, we'll be dealing with the double fertilization, different events, then post-fertilization, different stages. And in this episode, the first part of post-fertilization, that is endosperm, also will be discussed. That is our topic for this episode. So we can proceed. So I will share the screen, so it will be convenient for you. Then I think screen came. Now we can proceed. So sexual reproduction in flowering plants, fifth episode, mainly dealing with fertilization. After that, different stages, then endosperm development. So we can start. 5 main items we'll be discussing here that I written there. One is pollen tube, second pollen tube discovery, need, embryo pollen tube, importance of pollen tube. Third is double fertilization, product of double fertilization, zygote and endosperm, their ploidy. Main steps of post-fertilization events followed by endosperm. So we can start with the pollen tube. Microsperm mother cells undergo meiosis. 4 microspores will be formed. And when we take a single microspore, it will undergo mitosis. 2 unequal cells will be formed. One is large vegetative cell, other is small generative cell. Large vegetative cell or tube cell, which develops into pollen tube. So that is the basic starting point of pollen tube. Starts with the vegetative cell or tube cell. Pollen tube discovery is by an Italian scientist, G.B. Amici or Amici. In angiosperm, that is a popular question, male and female gametes both are non-motile. So in angiosperm, both the male gamete and female gamete both are non-motile. So male gamete should be carried to female spot. And that carrying event is done by pollen tube. And this is specifically an adaptation for land plants. Since no water will be available on land normally. And this phenomenon, with the help of a pollen tube, male gametes will be carried. It is called siphonogamy. So siphonogamy means male gametes will be carried through pollen tube. And remember what we studied in last episode, pollen tube growth is apical and it is chemotrophic in nature. Now, how the pollen tube enters into ovule? So pollen tube finally reaches into female site. It has to enter into ovule. How it is possible? 3 methods are available, which includes 1, through micropyle. That is a normal passage and phenomenon is called porogamy. Other is through chalasa. It is called chalasogamy. And the third one is through indegments. It is called mesogamy. So this is how the pollen tube enters into ovule. 3 passages are possible through micropyle, through chalasa, through indegments. Porogamy, chalasogamy, and mesogamy. Now, in these 3 methods, pollen tube enters into ovule. But how the pollen tube enters into embryo sac? So pollen tube can enter into the ovule by different methods, what we just observed. But the entry of pollen tube into female gametophyte or embryo sac is through micropylar end. So these two end rates should be kept in mind. Okay, so pollen tube finally enters into female gametophyte or embryo sac through micropylar end. And for this end ray, we can find synergids will direct the pollen tube in a correct pathway. So synergids will act as obturators. I think that idea is clear. Now, three famous NCERT diagrams. A and B showing how the pollen tube enters into the embryo sac, how the synergids will be helping for it, and what is the fate of synergid, etc. And the first two diagram where students will not be giving any importance. But remember, it is a theory question for labeling. Same time, it can be an entrance question for NEET exam labeling. NCERT diagram. Third one, that is an LS, longitudinal section, showing how the pollen tube grows. That also NCERT. So three diagrams, very prominent for exam point of view, both the theory and entrance. Third one, usually they're asking for theory exam also. I think three diagrams are clear. Try to label or learn how to label these three diagrams. Now we can proceed to next aspect. It is called double fertilization. Double fertilization is mainly concerned with the famous scientist Navashin. And this double fertilization, remember, it is a very unique feature for angiosperms, very unique to angiosperm. That itself is a question. And why it is called double fertilization? Because there are two events in it. One is called syngamy, other is called triple fusion. Now, what is syngamy? SYN, we already discussed, it means union. So syngamy, gametes will be fused. So syngamy, what happens is one male gamete fuses with egg. Remember, ploidy of the male gamete is N, ploidy of the egg is N. And the syngamy is initially worked out by a scientist called Strasburger. Syngamy, Strasburger. And haploid male gamete fuses with haploid egg. So total, it will be 2N, ploidy level, giving rise to zygote. That is syngamy. Second fusion is called triple fusion, where one male gamete fuses with the secondary nucleus. Again, male gamete haploid, secondary nucleus formed by the fusion of two polar nuclei, N plus N. So indirectly become 2N. Into this 2N, male gamete enters, so it becomes 3N. And this is concerned with the two famous scientists, Navashin and Gignard, triple fusion. And the product is triploid primary endosperm nucleus. That is how the primary endosperm nucleus in angiosperm, they become triploid. So after double fertilization, we can find two ploidy level we'll be getting. One is zygote with the 2N, other is primary endosperm nucleus with the 3N. The famous question about theory entrance. Even problems can be there. Now there is a diagram that is showing fertilized embryo sac, zygote and primary endosperm nucleus, and degenerating synergids, etc. I think that also clear. Now next is pollen tube and structures. So when we look onto pollen tube, we can find tube contain certain plugs. It is made up of callose. And it also contains two male gametes, and there will be a tube nucleus at the apex. And that is why it is suggested pollen tube growth is apical and it is chemotrophic. Now, next aspect for us is post-fertilization. Post-fertilization means, post means after an event. So initially pre-fertilization, gametogenesis, then pollination, then leading to fertilization. Now we are proceeding to after fertilization, what will happen? And this can be studied under 4 headings. One is endosperm development, because it is the nutritive tissue for developing embryo. Then embryo development. Then ovule develops into seed, ovary develops into fruit. So 4 stages we have to learn. First is, in this episode, we'll be dealing with the endosperm development. So first aspect is endosperm development. Ploidy of the endosperm in angiosperm is triploid. Questions will be there. A diploid male parent is crossed with a tetraploid female parent. What will be the zygotic ploidy? What will be the endosperm ploidy? This all I will put in after finishing this all chapters, a question series. Now, endosperm is formed after fertilization in angiosperms. But remember, in gymnosperm, it is before fertilization, it is haploid. Here it is after fertilization, and it is triploid. That is another usual question, either in plant kingdom or here. So angiosperm, triploid, as well as after fertilization, endosperm form. In gymnosperm, it is haploid and before fertilization. Now, functions of endosperm. Functions of endosperm, just like tapetum we discussed in anther, it is nutrition of developing embryo. And which is the mother cell of the endosperm, that is nothing but central cell that we have done in embryo sac. And in certain angiospermic families, endosperm will be absent. Typical example is Orchidaceae, belongs to monocots. And normally endosperm ploidy will be triploid, but rarely it can be diploid also. It is an abnormality. And this is, we can easily establish that is to an endosperm in a famous plant called Enothera lamarckiana, evening primrose, the famous plant in which Hugo de Vries worked, where the ploidy of the endosperm is diploid. Not in syllabus, but it is a frequent question for higher level exams. That is why I told. So ploidy of the endosperm in Enothera, evening primrose, it is diploid. And this Enothera belongs to a family Onagraceae, not in our syllabus. Now, next we have to proceed with how the endosperm, triploid endosperm develops. 3 methods are available. NCERT wise, basically 2. But a third method also available. The 3 we can cover. Diagrams are shown there. So first is free nuclear type. Here what happens is nucleus divides continuously. So many nucleus will be formed and a cell wall is laid at the end or last stage only cell wall is laid. That is why the term free nuclear type. So repeated nuclear division and the cell wall is laid at the last. And typical example for it is graminaceous members, rice, maize, and cotton. Cellular type, from the term itself, we can guess each or every nuclear division has got cytokinesis. So each one after division, cell wall will be formed. And it is commonly seen among balsam, petunia, datura. Many names are there, only 1 or 2. Now, third is helobial type. Helobial type originally not for us, but just for completion of the 3 types, just mentioning. Endosperm development is in between cellular and nuclear type, intermediate between cellular and nuclear type, not in syllabus, but just mentioned it. And how the helobial name came, it belongs to helobials of monocots. And one interesting fact, coconut has got free nuclear type of endosperm development and cellular type of endosperm development, since it contains liquid part, watery part, and a semi-solid part. So both prenuclear and cellular type can be seen in coconut. Exception. I think that part is clear. Now next is fate of endosperm. Endosperm, we know it is a nutritive tissue for developing embryo. And it can be 2 methods or 2 ways possible by which endosperm can be consumed. 1, complete consumption of endosperm during the development of embryo before seed germination. So endosperm will be completely consumed. And such seeds are called ex-albuminous or non-endospermic seeds. And it is popular among dicots. So ex-albuminous or non-endospermic seed, complete consumption of endosperm during the development of embryo before seed germinate. Now counter. Endosperm will be persisting in the mature seed. And since endosperm proteinaceous, it is staying there, it is otherwise called albuminous or endospermous seeds. Albuminous. Example is coconut, cereals, and castor. So 2 way, that is, endosperm will be completely consumed before or during the development of embryo before seed germinate. So before seed germination, endosperm will be finished. Ex-albuminous or non-endospermic. Example is groundnut, pea, etc. Endosperm will be persisting in the mature seed, as in the case of albuminous or endospermous seed, coconut, cereals, castor. After that, some questions has been provided and 5 theory, 5 entrance. Key also given there. And pass through these questions while doing 5 NEET type question. Don't look onto the key. After doing, compare it. And at the end of the chapters, all chapters, I will be putting series of questions with the key and even hints. So first we can finish with the theory part. So what all we discussed today? We are proceeding with the 5th episode of the sexual reproduction flowering plants, fertilization, double fertilization, then different aspect, pollen tube discovery, double fertilization, zygote, endosperm, ploidy, main steps of post-fertilization events, endosperm, etc. Then how that pollen tube develops, then entry of the pollen tube into ovule, 3 methods, then entry of pollen tube into embryo sac, only through micropyle. After that, we entered into double fertilization, how the zygote, how the primary endosperm nucleus, syngamy, triple fusion, ploidy level, how many nucleus involved, etc. After the diagram showing after fertilization what happens. Then a pollen tube growth, plugs, male gametes, tube nucleus, and how the pollen tube grows. It is apical and chemotrophic. Then post-fertilization, endosperm, embryo, ovule into seed, ovary into fruit, endosperm development. What is endosperm? What is a ploidy? And how it becomes 2N in certain families or certain plants. Then mode of development of endosperm, 3 methods, prenuclear, cellular, and helobial. From the term itself, we can guess how that development takes place. After that, fate of the endosperm, it is completely consumed, it is persisting. After that, 5 theory question and 5 NEET model question with the key. So normally one more episode we need because it is a big chapter. That is the last part of this chapter. So we can meet later. If you like this channel, please subscribe or like it, share it, and comment on it. Thank you. We can meet later.